Esther Kuehn holds a BSc in biology from the University of Munster and a master's degree in neuroscience from the University of Otago and Humboldt University in Berlin. She obtained her PhD in cognitive neuroscience at the Max Planck Institute in Leipzig in 2013. And she was a postdoctoral fellow at the University of College London before she joined DZNE Magdeburg in 2015 as a postdoctoral fellow, followed by leading a group titled Cortical Microstructure and in Health and Disease, which was, I believe, in Magdeburg. Esther, you can correct me if I'm wrong. She's now a professor for translational imaging of cortical microstructure at the Hertie Institute for Clinical Brain Research in Tübingen. And today we are super excited to be able to hear her talk about layer-specific models of cortical sensory aging. Welcome, Esther.
Thanks a lot for the nice introduction, and thanks a lot for the invitation. I'm actually very excited to present very novel work to you that is at the moment under review, and to discuss with you a layer-specific model on cortical sensory aging. So we have this at the moment in bioarchives, and just in case you wonder, the new version is having a different title. Just in the course of revision, we were encouraged to change the title, but cortical sensory aging is layer-specific, is the same work and just has more extensive analysis included. Just before I start, I would like to very much appreciate the great work that has been done by the two first shared co-authors, by Ping Lui and Juliane Döhler. They both contributed to this work in the course of their PhD thesis. They both now successfully submitted their thesis. And the work that I'm presenting on rather functional analysis was mainly done by Ping Lui, where Juliane had a bit more focus on the structural work that I will be presenting. So the overall question that was leading this research project was the question, how does aging and plasticity affect the layer architecture of the cortex? The topic today is translational imaging. So before we start any clinical research, patient research on layers, or we plan also to build new models on layer-specific pathology, we first need to sort of have an understanding of how the layer architecture changes in the course of healthy aging, and how it may be potentially changed also by effects of plasticity, because I think then we have the basis to then understand something about, for example, changes in pathology or neurodegeneration. And there's really not much knowledge there at the moment, neither in humans nor in animals, on actually the question of how aging and plasticity affect the layer architecture of the cortex. So this overall question we targeted using the somatosensory cortex as a model system. Specifically, we focused on area 3B, which is getting direct input from the mechanoreceptors or the thalamus from the skin. So this, in principle, a classical sensory input area, allows, therefore, to study things like sensory input, modulatory input, output dissociations. In addition, it is a key area for touch, haptics, motor control, body perception. It has clinical relevance for aging, neurological, neurodegenerative diseases, specifically the interaction between sensory motor cortex and the medial temporal lobe, where the insula is an important network for body memory. There's clinical relevance, for instance, for somatoform and trauma disorders. And important aspect here is also that the area has very good SNR in actually 7T images. So this is a good starting point, let's say, to look at some of the aspects that can, of course, then also be looked at in different areas of the cortex. So I will jump directly into the questions, because I think I have 15 minutes and you all know the layers. So I will directly start with, in principle, the first of the two questions that I would like to target. There's actually more on those in the study that I'm not focusing on today so much. However, I will try to explain a little bit about what we could find to answer two questions. The first is the architecture of input layer 4 sensitive to aging and sensory deprivation? I think it's an important question, because when we think about input-output pathways, of course, an important question is to think about what is actually about the input channel. is it sensitive to circumstances of aging and sensory deprivation? And there is not so much knowledge on this so far. There is this one study on mice where actually they had a trim condition, trimmed off the whiskers, and found myelination to reduce in layer 4 after time, and also showed changes in and the neural response to air puff stimulation to the snout. However, for example, in this study, there was no systematic investigation of all the layers. So it's not clear whether this was specific to the input layer or also other changes would show sensitivity, for example, to the input statistics. So how did we actually experimentally approach this first question? So we had a 70 MRI study conducted at the 17 Magdeburg. We measured altogether 2 cohorts of younger and older adults, where one cohort compared 20 young versus 20 older. That was the cohort mainly used for whatever you see in the next slides on structural imaging. And we had a cohort of 14 older and 14 younger and 13 older adults. Most of the work you see on the layer-specific changes are from this independent cohort. And we investigated a patient with congenital arm loss. We had MP2 range sequence of 0.5 millimeter isotropic. We measured with the same resolution QSM sequence. We used an 0.9 millimeter bold fMRI response or sequence for the functional imaging, and had to very briefly say it in principle a tactile stimulator that could apply passive touch to either all fingers or specific fingers. I come to the details a bit later. Then in order to actually say where is the input layer, to define the input layer, and I'm actually happy that Bob Turner is here today, because this is actually really work I'm referring here to his lab in Leipzig. So there was Diane Dinse who did an in vivo model of the primary somatosensory cortex, where she used the ex vivo work to define the layers, and then use the in vivo MRI data to say how can we actually now come back to the same layer division using the in vivo data. And we actually, and her data are here plotted in blue and our data are in black. And we showed that our in vivo profile nicely aligned with her model. We also used the same sequence actually, and could therefore actually take her definition that she developed based on the ex vivo data to define the differentiation between the superficial layers, the shown here in yellow, where we think is the middle layer shown in red, and the deep layers shown in blue. We had a second way of doing this, and this is all published in this last year's paper. That's why I'm having it explaining it very briefly only. But we also had actually PQSM data here extracted from the cortical depth, and showed the 2 peaks at the locations that should be corresponding to the inner and outer bilateral bands, which also corresponds to the locations of layer 4 and layer 5 as we defined them based on this method. So there are actually 2 independent ways of saying, of coming to the same conclusion where the specific layers may be located. So we took over this method also in here. The finger locations, so the question of where did we sample the data, was defined by using population receptive field modeling. Also, this was actually already introduced in much detail in an earlier paper by Peng in 2021. And we used this basically. So very briefly, the idea is that there is a model developed based on neurophysiological data that gives an estimate of how a bold response, or let's say generally a voxel-based response, should look like given a certain expected population response in response to, for example, visual or tactile stimulation. And then by fitting the data to the model, one can arrive at an estimate of where certain receptor fields are and how they're related to the fingers. This we use to localize the fingers. Then we combined in the present work, we combined this investigation with a mice investigation of younger mice that should be corresponding in their age to the younger adults, older mice that should be corresponding to our older adult age range between 6 and 80 years, and very senescent, very older mice that should be older than our older adults, let's say, to actually track some of the mechanisms a little bit more in detail. that we can only observe, let's say, more superficially in the MRI images. We used actually cytoarchitectonic analysis on postmortem data, but also in vivo 2 calcium imaging, and we used the 2 whiskers as the 2-finger analogy. In principle. So we had either 2 whisker stimulated an analogy to 2-finger stimulations, and we trimmed off 1 whisker and had 1 whisker stimulated an analogy to a single-finger stimulation condition. I'll also come to this a little bit later in more detail. So the first finding, the first, let's say, interesting result of this work is to say that aging increases and sensory deprivation decreases the thickness of input layer 4 in S1. So I explained to you before how we define the layers. And if you concentrate on the left-hand side of the slide, then you see that the red area that highlights the middle layer, where the input layer 4 is also located, is thicker in the lower panel in older adults compared to the upper panel showing the younger adults. Important here to say is that this difference is not only relative, but it's total. So you could say, because the cortex is thinning and aging anyway, potentially it's just that the layer 4 is relatively thicker, but it is also numerically thicker, irrespective of the cortical thinning. So that's important. So it was a numerically larger thickness, which was also significant in the middle layer in older adults. When you then focus on the middle panel on the upper left, you see that this is the extracted QT1 signal in the different compartments, and where the black arrow is, this is actually the T1 signal potentially reflecting myelination, showing also higher values in the input layer in older adults. In principle, it's plotted the way that the values more to the right are reflecting higher myelination. So it was interesting from the structural perspective. And when you now concentrate a bit more on the middle panel, this is then the bold signal analysis we did layer specifically, where in the lower panel, you first of all see, so each black line is an individual, whereas the red line is the average of older adults and the blue line is the average of younger adults. And first of all, you see that there's a shift to the right in older adults, which is reflecting generally overactivation in response to sensory stimulation. And this is actually something we also observed in the mice. So also in the mice using single neuron recordings, we see this sensory overactivation. So this is something we also expected to accompany aging. However, important is that we also saw a more pronounced sensory input signal in layer 4 in older adults. This was also significant when we normalized the data according to the general profile across the layers. So there was somehow a more pronounced sensory input signal in older adults, irrespective of the generally overactivation across layers in older adults. And then on the right-hand side is another interesting aspect, because we also had, of course, the amputees. So we had a model of a reduced sensory input. And here, what we could observe is, first of all, an overall cortical thinning in the S1 contralateral compared to ipsilateral to the missing arm. This is what you see here printed in red. This is expected. The face was somehow the control region where we didn't see this thinning because the face was not affected in this patient. Then the second part of the right panel is the interesting one, because we saw that the cortical thinning in the amputee is driven by the middle layer. So we see here that the largest difference actually between ipsilateral and contralateral hemisphere is the 0.11 millimeter difference in the middle layer, where the input layer is located. So what is interesting about those data is actually that it suggests that with aging, we actually have potentially a expansion of the input channel, potentially due to the increased sensory input that older adults, of course, get over the lifetime, also accompanied with a more pronounced sensory input signal in the layer 4. We now had the mice data where we first wanted to see actually, can we in principle see similar changes in mice, and could we actually understand a little bit more about the potential underlying mechanism? So here we had a myelin basic protein expression that we actually plotted here according to the layers. This is not usually how mice data are plotted, but we wanted to make it, let's say, comparable to how we plotted the human data. And you nicely see it in the average plot, that in principle you do see also the increase in the myelin basic protein in the middle layer 4. Please, for the moment, don't concentrate on the lower layer so much, because my next 2 slides will be on the lower layer. But if you could just concentrate on the input layer, on the red one, then you see that also in the mice, you actually do have this stronger myelination in the older mice in the input layer, which is also significant. And interestingly, we see a degradation actually in the very old mice. So we think that there's potentially 2 mechanisms coming into play. One is a plasticity-based mechanism, where actually more input over the individual lifespan is increasing the microstructural complexity, the myelination specifically, but also the size of the input layer, whereas in this case, neurodegeneration, but also in the plasticity case, a reduced sensory input is shrinking this layer. Just having a look how much time is left for the second question. It's already 14 minutes. So I'm trying to be very quick with the second point, not to go over time.
You're fine.
I'm fine? Okay, because I was told 15 to 20 minutes, so yeah, I try in the next 5 minutes. So the second question is actually, we know that the cortex is thinning with aging. This is a very established finding that has actually been reported many times. When I'm now telling you that the input layer is in principle thickening with age, also in total numerical terms, then of course the question is which layer drives the age-related cortical thinning, right? Because this is actually the mechanism also that may be related to neurodegeneration that we may want to understand. And here, our data show that the age-related cortical thinning that we show also over the full cortex is driven by deep layer thinning. So when you concentrate first on the left-hand side, then you see that the blue, that is the inner layer that is highlighted in blue, is actually thinner in the older adults compared to the younger adults. And this was also statistically significant. So the first insight was, okay, the overall cortical thinning that we find is actually really triggered by the deep layer. That may mean that whatever we see in terms of degeneration related changes in aging may actually be strongly driven by the deep layers. And therefore, of course, it may be making sense to concentrate on the deep layers a little bit more. So first of all, we were investigating the microstructure of the deep layer, again, using the quantitative T1 as a readout. And there you also see in the middle panel with the black arrow highlighted that also in the deep layer, there's an increase, let's say, in myelination, so a decrease in the QT1 signal, which is first of all, of course, a little bit surprising, because you may kind of think that the cortical thinning or deep layer thinning is sort of accompanied by a reduction in the microstructural or in, let's say, in the marker for cortical myelination, right? So we were therefore a little bit more closely looking at the data. So what we were suggesting, our own model, was that the deep layers are actually mediating cortical, let's say, activation. Specifically, they're mediating a functional inhibition between co-activated areas. And we had, I told you at the beginning, we had this a paradigm where we either had 1 finger stimulated or 2 fingers stimulated. And therefore, we could actually measure a kind of established marker for cortical inhibition, which is to compare single versus double finger stimulation to see how much is actually an activation reducing in the double finger stimulation case compared to single fingers. In the table below, you see the values using 2 different metrics to quantify this. And the higher the value, let's say, to simplify it, the more the inhibition. And surprisingly, you see the values are a little bit higher for older adults. And the base factor, however, is relatively low. So there's no strong evidence for a difference, but if at all, there's a tendency for older adults showing more inhibition. Also, of course, overall a surprising finding, because we know that GABAergic inhibition is reducing in aging. And here, actually, again, the mice data could help us to understand this a little bit better. So I already told you before, showed you in the previous slide, that also in the mice, we did have this myelin basic protein extension, let's say, or a higher amount of myelin-basic protein tracking in the older mice compared to the younger mice. This is the bottom row of the left panel. And you have this further reduction again in the very old mice. So in principle, we confirm, first of all, this increased myelination that I showed you here in the humans by having also this shown in terms of a myelin-basic protein expression increase. Interesting is now that we further show that this myelin-basic protein expression is correlating with PP plus neuron density. And this PP plus neuron is a marker that or PP plus neurons often encompass interneurons. So in principle, it means that the myelin-basic protein expression is very much driven by inhibitory neurons. And this, again, is then interesting looking at the functional results in the mice, where you see that using the whisker stimulation, where we have the single or double whisker stimulation, or the by comparing the response, you can actually identify additive neurons and neurons with a reduced profile or a suppressed profile, really on the individual neuron basis. And there you can really track in different layers which neuron shows which response profile in the younger and older mice. And here actually, and you see the inner ring is the inner layer and the outer ring is the outer layers. What you see is that in principle, the strongest difference is in the reduced profile, that older adults in the outer layer have a higher amount of neurons that show a reduced response profile doing single versus double whisker stimulation. Whereas the suppressed profile is very much the same in younger and older. And this is in principle also, again, aligning with what I showed you here, where we actually had no strong difference between the inhibitory responses between younger and older adults, but if at all, a tendency for the older adults to show more suppression. This is actually also seen in the mice. So what we think could be happening is that really this PP plus neurons are a compensatory mechanism that actually helps at this age stage to compensate for the GABAergic loss, but that in very old mice, where we actually don't have the equivalent in humans, we don't have the very old humans here in the sample, this would then break down potentially and then actually explain then the strong age-related functional effects that we see then in the very older age range. So that was very quick, and I hope I could come across with some of the interesting results we had in the study. This is a sort of layer model of sensory aging that we try to summarize those different aspects: the deep layer thinning, the input layer enlargement, let's say schematically. But I would rather like to take one last minute to discuss with you some considerations that I think come with those findings. So I think the first thing is that when the thickness of the input layer 4 is dependent on sensory input statistics, I think it introduces input layer thickness as a very interesting and novel marker of input-driven cortical plasticity that I think could be investigated in many other contexts in the course of plasticity and learning. The second aspect is given middle layers are larger and deep layers seem to be thinner in older adults. Across-layer cortical thickness does not seem to be an appropriate metric to describe cortical degeneration and plasticity, because when you measure the mean and you have some of the parts enlarging and some of the parts shrinking, the mean is not informative, right? I think that's important because cortical thickness is still the major readout actually used to classify aging and degeneration. So we may have to think about more precise metrics in this respect. And the last point is, I think for me, it was very interesting to see in mice this correlation. First of all, also the increase in the myelin-basic protein expression, mirroring the results in humans, but also the correlation with the PP plus cell density. I think it introduces potentially a novel interpretation also of how we interpret QT1 signals in humans, also in the course of aging or plasticity. So I think for me, this is also a really interesting novel marker we have to think about more. And this is in principle already my last slide. I see already the first questions. Thanks a lot, first of all, to my collaborators, most of all, Janelle Pakan, because she and her lab were conducting the mice analysis, and it was enormous effort for the lab to actually do this, because there was no major funding to them. They all did it on the side, and there was major, really effort to contribute to this work in this way. So there's a big thanks to the lab of Janelle. And of course, thanks to full lab for supporting those those studies and also introducing novel methods in the course of this. And thank you very much for your attention. And now I already see,
Amazing.
yeah, are you moderating the session, right? Yeah.
Yeah. So I will just say very quickly that normally our format is that everyone kind of has one of their talks, and then there's a larger discussion afterwards as kind of the generalized theme, etc. Bob, if there are burning questions for Esther, we can do that while we're setting up this talk. But we'll save most of the questions, I think, for the general discussion.
Just a comment about quantitative T1 as a marker of albumin cell density. Esther, have you seen the paper by Annika Alcamada, published about 4 years ago now, showing sections of the human brain where you've got myelin stain and albumin stain? I've been looking at those, really interesting. They don't match perfectly, which is very interesting. I just thought I'd get that one in. You'd love my talk coming up in Amsterdam. Maybe I'll send you the slides.
Yeah, please. Yeah, we see each other next week, right? Yeah.
Yeah.